So for tutorial 1, this is basically an introduction to BB101 and the topics which BB101, and for the first half of the semester, like the topics which will be focused on is molecular and cellular biology. So for today, like we will be talking about the cell. So what is a cell? A cell is the functional unit of life, and it is also the smallest unit of our body, like the smallest unit which we can see under a simple microscope is the cell. So there are two kinds of cells. One is the prokaryotic cell and one is an eukaryotic cell. So prokaryotic cells are the more primitive kind of cells. These are found in bacteria, and eukaryotic cells are evolutionary more advanced, and they are found in higher organisms such as plants and animals and humans. So this is a general structure of a prokaryotic cell. So here we have the picture of a bacterial cell. Okay. So the distinguishing features of the prokaryotic cells are that on the surface there are thread-like projections called fimbriae, and in the nuclear part, like the nuclear part which contains the genetic information, is called the nucleoid. Like for an eukaryotic cell, we call it a nucleus, but for a prokaryotic cell, we call it a nucleoid. I'm coming to the difference between a nucleoid and a nucleus later on, but just for now, remember that the genetic material of the prokaryotic cell is concentrated in a region of the cell called a nucleoid. Also, there are some organelles which are involved in synthesizing proteins. These are called the ribosomes. And the outer covering of the cell is called the plasma membrane. Outside the plasma membrane, there is a rigid structure which is called the cell wall. Now, cell wall is present in certain kinds of eukaryotic cells as well. Like for example, the plant cells, they have a cell wall as well as the fungal cells. So cell wall is not an unique feature of the prokaryotic cells. Also, some of the prokaryotic cells, they outside the cell wall, they have a structure called a capsule, which is present on the outermost part, just like behind the fimbriae. And this capsule, it can be a jelly-like substance which can slime. This is feature of certain kind of bacteria. Also, in addition to the fimbriae, some prokaryotic cells, they can have long thread-like elongations. Like the fimbriae, they are smaller elongations, but flagella, these are longer projections like this. Like they are like tails. Like they help the bacteria to move forward. Okay. Okay, now this is the structure of an eukaryotic cell. Now just by looking at it, we can see that the structure is much more complex than that of a prokaryotic cell. So you remember, like I said, that the genetic material of a prokaryotic cell is concentrated in a region called a nucleoid. And in case of a eukaryotic cell, we call it the nucleus. So the difference between the nucleoid and a nucleus is that the nucleus has a thing called a nuclear membrane. So the entire genetic material, that is the chromosomes, they are present inside the cell. They're surrounded by a membrane, which we call the nuclear envelope or the nuclear membrane. And because of this, the nucleus is a well-defined structure. Like when you look at a eukaryotic cell under a microscope, you can distinctly see that yes, a part of the cell, there is a thing called a nucleus is present there. But in case of a prokaryotic cell, this nucleoid, it does not have anything such as a nuclear envelope or a nuclear membrane. Like it's kind of a diffuse structure. Like the genetic material, the chromosomes, they're spread out over a certain region, but there is no covering membrane. Like it's more or less, it is diffused around in the cytoplasm. It's concentrated in a certain part of the cytoplasm and is diffused. So when you look at a prokaryotic cell under a microscope, the nucleoid or the genetic material part is not exactly well defined. So that's the difference between a nucleoid and a nucleus. Also, in a nucleoid, there is no such thing as a nucleolus. Now, what is a nucleolus? Like in an eukaryotic cell, you can find a thing called nucleolus inside the nucleus. So a nucleolus is a non-membranous structure involved in the production of ribosomes. So eukaryotic cell, it has a lot more genetic material than a prokaryotic cell, and because of this higher amount of genetic material, a lot more things go on inside the nucleus of a eukaryotic cell as compared to a prokaryotic cell. So an eukaryotic cell nucleus requires more protein than a prokaryotic cell. That is why the eukaryotic cell has a separated eukaryotic cell nucleus has a separate entity for protein synthesis for its own. This is called the nucleolus. So this nucleolus is distinct from the ribosomes, which are involved in protein synthesis for the cell as a whole. Okay. So also inside the nucleus of a eukaryotic cell, obviously there is the chromatin or the genetic material proper. What are the common features between the eukaryotic cell and the prokaryotic cell? So here also we have the outer covering, which is the plasma membrane. We also have the ribosomes, which are for the protein synthesis. We also have small projections on the surface, which are called here the microvilli. Microvilli are not present in all the cells. They are present in some kind of eukaryotic cells. And they're more or less comparable to the fimbriae. Here also, like there are some cells, they might have a flagellum. Not all eukaryotic cells have a flagellum, some do. So these are the common features. And you can see that majority, there are a lot of differences between eukaryotic cells and prokaryotic cells. Like namely the eukaryotic cells, like there is a lot more compartmentalization and there are structures called organelles. Like for prokaryotic cells, we did not have anything called organelles. It was just the genetic material in the nucleoid, the ribosomes for protein synthesis, and the outer covering of the cell, and that's it. But here we have a bunch of other things inside the cell apart from all these things. And these are the things that we call the organelles. So what are the organelles? I'll go one by one. So one is the Golgi apparatus. This one. Golgi apparatus you can identify as this kind of a like coil structure which is folded on itself multiple times. And it's involved in synthesis of different kind of compounds inside the cell. Like different kinds of compounds like lipids, and they produce different, like other than proteins, they are involved in synthesis of other molecules. Then there are organelles called the lysosome. Now the lysosomes, they are involved in the cell defense. Like they produce certain substances which help defend the cell from bacteria, from other microbes. And they also are involved in destruction of macromolecules, when they're excess or when they have already reached their expiry date. And one of the most important organelles in a eukaryotic cell is the mitochondrion. Now, I think everybody has heard of something called this mitochondria. It's the organelle which is involved in cellular respiration, and it produces energy or ATP, and it's called the powerhouse of the cell. And other organelles here are, one is something called a peroxisome. It produces hydrogen peroxide, and hydrogen peroxide is hydrogen peroxide destroys free radicals. In the cytoskeleton is part inside the cell which is involved in the intracellular transfer, like transfer of different materials, say from the cell surface to the cell interior, and they are made of microfilaments and microtubules. Also, there is something called a centrosome. The centrosome is involved in the cellular cell division. It plays a role when the cell is going to divide. And also there is something called an endoplasmic reticulum, which is involved in protein synthesis. Like this is also quite a folded structure like this, and is attached to the nucleus and is studied with ribosomes. Like in case of the prokaryotic cells, the ribosomes, they are freely floating around in the cytoplasm. But in case of an eukaryotic cell, most of the ribosomes, they are embedded in the endoplasmic reticulum, and very few are like floating around. So endoplasmic reticulum, they are of two types. Like one is the rough endoplasmic reticulum, which is the ones which are studied with the cytoplasm, studied with the ribosomes, and they are involved in the protein synthesis. And the smooth endoplasmic reticulum, and this is comparatively less in quantity as compared to the rough. And the smooth endoplasmic reticulum, it's like involved in synthesis of different compounds other than the proteins. Okay, so this is a same picture like of the before, like just a bit more well defined. Here you can see like it's better like shown here, like the Golgi apparatus looks like this. The mitochondrion is like this. And this is basically a plant cell they're showing here. So in the previous picture of the eukaryotic cell, like you can see that this may be an animal cell, okay, because there is no cell wall here. It might be this. And this might be, it's a plant cell, because outside the cell membrane, there is this thing called the cell wall. And in case of the animal cells, like there is no such thing called a cell wall, it's just the cell membrane. But in case of a plant cell, outside the cell membrane, you have the cell wall, which is similar to that in like a cell wall is also present in case of the prokaryotes, but the composition of the two are different. Like the prokaryotic cell wall is different from the eukaryotic cell wall. And in case of the plant cells, there is an extra organelle which is not there in the animal cells, which is the chloroplast. And this chloroplast is involved in the photosynthesis process. So biological macromolecules, like there are 4 kinds of organic macromolecules which form the building blocks of life, and these are namely the nucleic acids, proteins, lipids, and carbohydrates. So let's go one by one. So what are nucleic acids? So nucleic acids, they form the genetic material of the cell. So genetic material is for hereditary, not only for hereditary purpose, it controls everything in our life. Because what happens, like at the core of every cell, there is something called a DNA. The DNA, the famous double helical structure, which all of you have seen. So this DNA, it's called transcription, from which there is a molecule called mRNA, which gets produced from DNA. Now this mRNA molecule, it stands for, RNA stands for ribonucleic acid, and M stands for mitochondria. So this mitochondria contains
Yes, Nishta.
I'm unable to hear.
Yeah, there is some problem. We are sorting it out.
Okay, okay, yeah.
I'm sorry, I think I got disconnected. Yeah. So am I back now? Am I audible to everybody? Diksha?
Yes, you are.
Okay. So, okay. So this mRNA molecule, it comes out into the cytoplasm. And then the ribosomes, which I said are involved in the protein synthesis, they wind around this mRNA molecule in a fashion like this. Like there are two ribosome molecules. One ribosome molecule which has two subunits. One is a larger subunit and one is a small subunit. So these go and envelop the mRNA in this kind of fashion. Like the mRNA is like a thread between the two units of the ribosome. It forms a structure like this, and then the ribosome, it undergoes then this entire process. This is called translation, in which with the help of the ribosomes, protein is being formed from the mRNA. So we can see that the original molecule, which is the DNA, which is our main genetic material, it gets converted into something called an mRNA by the process of transcription. And then this mRNA, it gets converted to proteins by the process of translation. So what happens, like DNA, our genetic material, it gets converted to proteins. The ultimate result of DNA is protein. So in this way, the DNA expresses itself. So DNA, it helps to code for different proteins, and these proteins, they carry out different functions in our body. So this is the structure, the general structure of a nucleic acid like DNA. So there is a sugar, a 5-carbon sugar. There is a nitrogenous base, and there is a phosphate group. So what is the central dogma of cell? Like the thing which I just described, like there is DNA, DNA gets converted to RNA, and RNA gets converted to protein. This is called the central dogma of cell or the central dogma of life. So DNA, like the process of transcription, it occurs inside the nucleus, it produces the RNA. Then the RNA, it comes out into the cytoplasm and it forms protein by the process of translation. And this translation occurs inside the cytoplasm with the help of ribosomes. Now this is the general structure of DNA. So DNA, it's in the form of this, like there is a double helical structure, and this DNA double helix is wound around certain proteins called histones, and this histone plus DNA combination, this is tightly wound around multiple times, and it gives rise to this kind of structure which we call a chromatin. And now chromatin is, it's not usually present in this kind of X-shaped fashion. Like this chromosome which you are seeing here, this double structure, like this X-shaped structure, is present in the mitosis stage of cell division. I'll get to that later. Usually it's just present, just half of this. Like mentally imagine that this is half, like just one short arm here and one long arm here. And like these arms, these are called the chromatids. And during cell division, like the chromatids, they get doubled. So this leads to the formation of this X-shaped structure. Anyway, so this coiled DNA plus histone combination, like which we can see
Shatirupa.
under.
Good morning, Sanjeev here.
Yes, sir.
Yeah, hi. Good morning, students. So, just also, ask them to ask questions in case they had some doubts in the previous lecture for some cell organelle. You're explaining very well, but I think in case they want to clarify some doubts, also give them opportunity to ask questions. Okay.
All right.
Yeah, continue.
Okay, so the two arms of the chromosomes, these are called the chromatids. And this point where they are joined together, this is called the centromere. So this is the structure which you can see under the microscope. Like you can't usually see this double helical structure or this histone proteins under a regular microscope. You need a much more powerful microscope for that. But this is the structure which you can, this chromosome structure is the thing which you can see under a microscope. So this is again a generalized structure of the DNA. So we have a 5-carbon sugar, which is we call the deoxyribose sugar. There is this nitrogenous base. Now the nitrogenous base, it can be of two types. Like one is can be a single ring or a double ring. And I'll get to that later. Like these are called the purines and the pyrimidines. And also there is this thing called a phosphate group. Now, what is base pairing? Like a unique feature of this DNA is that I already said, see here, that is double-stranded. So how does this double-stranded thing formation take place? Like here in this image, you can see that there is G bonding with C, there is A bonding with T. So what does this mean? So G, like the names of the nucleotides, okay, the names of the nucleotides, A means adenosine, T means thymidine, G is guanosine, and C is cytosine. So this is a dictum, like G always binds with C, A always binds with T. If there is any deviation from this, that means it's an abnormal DNA, it is some kind of mutation. And that will eventually be destroyed by the nucleus itself. So G always binds with C with the help of 3 hydrogen bonds. A binds with T with the help of 2 hydrogen bonds. So A and Diksha, can you just confirm this with me? So A and G, these are the purines, right? And C and T are the pyrimidines.
Yeah, you're right.
Okay, so A and G, these have their nitrogenous base. It's a bigger in size, like it has 2 rings, and these are called the purines. And C and T, their nitrogenous base has 1 ring, and these are called the pyrimidines. So a purine always binds with a pyrimidine. This is the thing. This is the way it happens. So the DNA sequence, like even in case, so the DNA sequence, it like is unique for every individual. So even though it's unique for every individual, doesn't mean that none of us have any similarities. So even in case of unrelated people, a certain fraction of our DNA is same. That's why people, even though we are different, we do look alike, right? Like we have 2 eyes, we have 2 arms, we have 2 legs. We have hair, we have eyebrows. So because of the certain, like a major fraction of the DNA in case of all human beings, it is same. And actually a very small percentage is different, and that is what makes the DNA unique for every people. So for parents and offspring also, like our genetic, like our DNA, it's more similar to our parents and with our siblings than with unrelated people. And also from an evolutionary perspective, like man, we have evolved from monkeys or chimpanzees. So a lot of our DNA sequence, like it is similar to that of chimpanzees, and that is how scientists they first discovered that yes, we did evolve from chimpanzees, and that's how we are related to them. And even in something like something like cabbage, like how are some we share some of our DNA sequence with certain some things like cabbage also, like this fruit, this vegetable. So DNA sequence is very very interesting, very fascinating that, we share DNA with unrelated people, we share DNA with an animal like a chimpanzee, and we share DNA with something like cabbage also. So it's very unique and fascinating. So then ribonucleic acid, like I said, this ribonucleic acid is produced by transcription from DNA. Now the structural difference between RNA and DNA is from just looking at this, you can probably not guess what the difference is. It's mostly same, but the only difference is that in case of DNA, the sugar is deoxyribose, and in case of RNA, the sugar is ribose. So at this group, the 2' group here, here there is an OH group, whereas in case of the DNA, this part, it does not have an OH group, it has an H, only hydrogen. So that's why, because this oxygen is absent in this 2' position in DNA, that's why we call that deoxyribose. But since this oxygen is present here, this, that's why this is not deoxy, this is just RNA, ribonucleic acid. And the bases, the bases, they are mostly the same, but in case of the T or thymidine, which we had in DNA, we have uracil here.
Sorry to interrupt, but Vaibhav asked a doubt regarding that can we make a DNA by combining two RNA? So Vaibhav, here is the answer that DNA and RNA both are different. So this is the main difference that Shatrupa recently explained regarding the RNA. So no, we can't make the DNA by combining the two RNAs. I hope you get the reason.
Yeah, so why can't we make DNA by combining two RNAs? So the one reason is definitely this, the sugar itself is different, although structurally it's quite similar, but actually it's different. There is one oxygen group here, there is no oxygen group in DNA. So even though this thing looks this difference seems very minor on paper, but actually in real life, just presence of this oxygen group, it makes a huge difference. And also, like I said, we have uracil in place of thymidine, we have thymidine in case of DNA. So in DNA, we showed you here, we have this T, and this A, it always binds with T. And because of this binding here, G with C, A and T, this is what forms this, this backbone of this double stranded thing. You see, these two connecting things here, these are these connections, G and C, A and T. So if we don't have T in RNA, instead of T, we have uracil. And now this uracil, it can't go and bind with A because of certain structural issues. So because of this, we can't have this double-stranded RNA. But, there are some double-stranded RNA viruses, some double-stranded RNA, some double-stranded RNA viruses. So, in their case, the bases, they are different. The structure of the RNA molecule itself is different. So I'm not really going into that. It's going to become complicated. So just for the purpose of a general eukaryotic cell, general human cell, just know that, two RNA molecules, they cannot be combined to form, a DNA or form a double-stranded RNA. So what are the types of RNA? So one is the messenger RNA, which I already talked about. So this one, it goes and sits between the two units of the ribosome to form the proteins. And also there is something called a ribosomal RNA. Now this ribosomal RNA, it's a part of the ribosome itself. And another thing, this is the transfer RNA. And it has this kind of a unique clover, we call this a clover leaf structure. And this is involved in the protein synthesis also. In the protein synthesis, we have to join the individual amino acids one after the other. So this transfer RNA, it's involved in bringing the individual amino acids together to form a long polypeptide chain. So this is a question for you. Which of among DNA or RNA, which one do you think is going to be more stable? Just write it out in the chat box. What do you think? So Diksha, what's the general consensus among the people? Which one is more stable, DNA or RNA?
DNA.
Is anyone
Answering it DNA.
okay? So you are right. DNA is more stable. Now, why is that? Because DNA is, is a double-stranded structure. So structurally, it's obvious that it's going to be more stable. And RNA, it's a single strand. So because of this single strandedness, it's much more prone to mutations. So in DNA, like I said, A has to bind with T, G has to bind with C. So because of this binding specificity of the nitrogenous bases, like there's very little scope for mistakes. And even if some mistake occurs, it leads to such a drastic change in structure that the cell will definitely figure out, yeah, something has happened here, some mutation has happened here, let me destroy this piece of DNA, or is going to cause problems for the cell. But with RNA, like in case some change happens, like in case of a particular base, some other base has been inserted there, like there's no drastic structural difference as such. So it's difficult for the cell to figure out that a mutation or some change has happened in the RNA. So that is why RNA is much more prone to mutations as compared to DNA. So an example of this is the COVID-19 virus. So COVID-19 is an RNA virus, and every few months, like every 2-3 months, you can hear about some new variant which is coming up. So just ever since COVID-19 was discovered, like I guess it was in the beginning of 2020, so it hasn't even been 2 years yet, and already like there are so many variants which have come up: alpha, beta, gamma, delta, mu, etc. Right now, Omicron variant is going on. So it's because of this, because COVID-19 is an RNA virus. That's why it's mutating so rapidly, and because of these mutations, is producing so many different kinds of these variants. So instead of if COVID-19 was a DNA virus, suppose instead of being an RNA virus, if it was a DNA virus, then it would not have undergone so many mutations. Then so many variants would not have come up, and we could have controlled it more easily. But unfortunately, it's an RNA virus. It mutates more rapidly. So what can we do? So what are proteins? Like I showed you in the image, like DNA, it gives rise to RNA, and then this RNA, it gives rise to proteins. So proteins are very large molecules, and they are formed of smaller molecules which are called amino acids. And all the functions of the cells, they are eventually carried out by the proteins. So the proteins, they are also called the workhorses of the cell. And like I said, the sequence is determined by the DNA. Like the DNA is like it synthesizes the mRNA, and according to the sequence of the mRNA, different amino acids come and join together, and they form the protein. And thus the structure of the protein is like according to the different proteins have different structures. So there are 4 levels of protein structure. One is the primary structure, which is the linear chain of amino acids. Like these structures, they're like present in all the proteins. Okay, so the primary structure, like it just has like this long thread of amino acids one after the other. It's very easy. Like the most simple structure of the protein is this. Then the next level of protein is the secondary structure. So this primary structure, this thread, it gets wound on itself to form something called an alpha helix, which is this alpha helical structure, which and hydrogen bonds are formed between the different amino acids, or something called a beta pleated sheet, which is like some kind of pleating is there. Like you have seen pleats in women wearing sari, something like that. Like it looks like a pleat. So this is what happens when the primary structure, it folds on itself. It gives rise to an alpha helix or a beta pleated sheet. So then this secondary structure, it gets further folded on itself to form something called a tertiary structure. Now the structure is more complex than that. It's a 3-dimensional thing. So primary structure and secondary structure, these were two-dimensional, and tertiary structure is 3-dimensional. And for some proteins which have only one chain, like one single chain of polypeptides that folds on itself to give the secondary structure, that folds on itself to give the tertiary structure. This is it. This is the final structure. But for some proteins which have multiple subunits, in that case this is one subunit, another subunit comes and binds to it, and it gives rise to this huge kind of protein. In that case it gives rise to something called a quaternary structure. So quaternary structure is not there for all the proteins. It's only there for some proteins like transthyretin or hemoglobin. So protein structure prediction in a certain softwares have been developed which can help us in predicting what the protein structure of the protein is. Like we are everyday scientists are discovering new proteins, and we need this AI methods, they help us to predict what is going to be the structure of the protein based on just the amino acid sequence. So this is just a brief overview of the protein function. Like proteins, they carry out a wide variety of functions. Like all the enzymes in our body, they are proteins. Like some store for proteins also function storage functions, such as casein in milk or ovalbumin in the eggs. Like all the hormones or the hormones in our body, they are proteins such as like insulin. Then motor proteins, there's these motor proteins such as actin and myosin, they are involved in muscle tissue, muscle contraction. Then the proteins, they are involved in the body defense, like antibodies. These are protein molecules. Then there are transport proteins such as hemoglobin. This helps in transport of oxygen. There are receptor proteins such as these receptors which are involved in neurotransmitter signaling or something like that. Then obviously there are structural proteins. They give help in structural rigidity of the cell, like keratin, collagen, elastin, etc. And the final group of macromolecules I'm going to talk about are the carbohydrates. So carbohydrates, they are present in both monomer and polymer form. So these are also carbohydrates are also called sugars. So the monomers are the more readily available forms of energy, and the polymers, these are the more complex forms of energy. So all the things which we eat in daily life, like rice, roti, wheat, pasta, anything, those are all the polymers. Like they are the polymers of carbohydrates. And the monomers, these are the simple sugar. Monomers or simple sugars, they are they are glucose or fructose, stuff like that. So monomers is basically what you see in stuff like glucon-D, that contains only glucose. But all the solid things which you eat in daily life, like roti or rice, they are the polymers of carbohydrates. So what happens in our body is that we ingest the polymers, like suppose we are eating rice or roti. It undergoes the process of digestion, and in digestion this occurs something called hydrolysis. And by the process of this hydrolysis, the polymers, they get converted into the monomeric form. And these monomers, they can be absorbed by our intestine, and they ultimately go on to carry out different functions in our body. Also there are lipids. Now lipids, these are also the fat molecules. So the general structure is that this kind of looks like a matchstick with two tails. So this red thing, this is called the head, and these two, like these yellow things, these are called the tails. So this is the structure of the cell membrane. So it has the hydrophilic head. This red thing is the hydrophilic head, and the yellow things, these are the hydrophobic tails. So that's why the heads, they are directed outside of the cell and directed towards the interior of the cell. And this hydrophobic tail part, this is the part which is actually embedded inside the membrane. Okay, so these lipids, they have long chains of carbons, and they usually have a range from 4 carbons to 36 carbons. And the lipids, they usually play two kinds of roles. One is the structural role, like the phospholipids, like these are the phospholipids in the cell membrane, and they also form the.
Just a reminder, we are running short of time, so please be.
Okay, yeah, I know. And they also form energy storage, like the triglycerides. So lipids are storage, like we consume, like when we eat, like we eat the carbohydrates, fat, and protein. So which one has more energy? So 1 gram of carbohydrates has 4 calories, and 1 gram of fat has 9 calories. So it's obvious that fat contains much more energy than carbohydrates. And regarding this, general biology, recent Nobel Prize winners are David Julius and Ardem Patapoutian, and they won the Nobel Prize for the discovery of certain receptors for temperature and touch. So I'll give some examples of real life applications of biology. Like one is artificial retina. Like using different engineering methods, we have developed this retina, which is it's completely man-made. It's an artificial retina. It gets implanted into the eye by the surgeon, and it is acts as a replacement of the original retina. Also, certain devices have come up which help us to measure, different body parameters such as our heart rate. Like this is in case of the Apple Watch is there. Apple Watch has to measure the heart rate, pulse, stuff like that. And there's also something called a cardiogram. Like this cardiogram is a certain company which has developed this technology to measure the heart rate using different electronic devices such as Android Wear, Fitbit, or Apple Watch. And in the future also, like we can use maybe these smartwatches to measure the glucose, blood pressure, and oxygen levels also. So partnership between biology and technology. So in the future, like as we are moving forward in time, more there is more intersection of biology and technology which is going on, and this is leading to a lot of societal impact. Like we are developing much more newer technologies such as the artificial retina, artificial eye. Someday we might develop artificial organs also with the help of technology. So this intersection with technology and biology is very crucial for the progress of the human race. Okay, so I'm done with my part. So Diksha, you can take it forward from here.
Yeah, okay, so I'm just sharing my slides. Is it visible?
Yes.
Okay, so here we come to the discussion session. So we have some interesting questions you can answer. So you can raise your hand and Shatropa will ask. Okay. So the first question is, first of all, the one of the most interesting fact is that we have one kind of cell that are enucleated. So RBCs are the only cells that don't have any nucleus. So why do RBCs don't have any nucleus? Is it related to its function? Can anybody guess? And one more question is that what is the shape of the RBCs? I would like to have some guesses.
Diksha, how do I unmute somebody here? Like some people have raised their hands.
If they have raised their hand, you just ask their name. You just ask them to.
Okay, so they can unmute from their end only.
Yeah.
Okay, sure. Nishtha Agarwal, you can go.
The shape is like the poppin shape. It's there's a depression in the middle, and
Okay.
the outside it's bulging, and it is so because it can transfer. It takes less space, so there's ease of movement.
I lost your voice, Nishta.
Where did you hear?
Shitrupa, can you hear?
No. Nishtha, you can type it out in the chat box. Okay, V Gayatri, you can go.
Uh, Actually, I don't remember the exact name of the shape of the cell, but the purpose of that shape is that so that it can squeeze through the blood vessels and all.
Yes, you are right. And so why these RBCs are enucleated? Is it related to its function?
Enucleated, so that since there is no nucleus, it's a pathway. Is it because it can travel more easier through the blood vessel, and it can due to large surface area?
Actually, you are partially right. It is because of the shape that it can travel easily, but it is enucleated because the nucleus gets out of the RBC, so it can make more space to accommodate the hemoglobin. So what does the hemoglobin do? Hemoglobin carries the oxygen, so it can carry the oxygen efficiently to all the organs of the body. Okay, so that's why it is enucleated. So the next question comes that after boiling the egg, the egg yolk becomes green. So why is it so? It is already written, but you can also explain.
Okay,
Okay.
Nishtha, you want to say something? Okay, Gayatri.
I
Gayatri,
forgot to down the hand.
okay, like your hand is still up, that's why I called your name.
So I'll explain. So what happens when we boil the egg? So the egg contain hydrogen and sulfide. So when we heat it, so hydrogen and sulfide reacts. So this hydrogen sulfide reacts. It forms hydrogen sulfide, and in the presence of iron, Fe2 plus ion, it forms iron sulfide, which is a green colored compound. So that's why it becomes green. So the next question comes that we know that fireflies produce light. So how fireflies produce light? And we know that light emits heat. So if then how do fireflies survive? Can anybody guess? Like why do fireflies produce light? Not exactly why. How do they produce light? So it is basically due to the presence of an enzyme luciferase. So this luciferase enzyme, what it does, this enzyme carries out the reaction. It converts the luciferase, it is a protein, into a light producing. It carries out a light producing reaction. So when this luciferase enzyme acts on luciferase in the presence of calcium, ATP, and oxygen, so it forms a light producing compound. So that's how light is produced. And in fireflies, this light is a cold light. So that is without a lot of energy being lost, heat is produced. Okay. So the next question comes the biomimicry. So do you know what is biomimicry? Anyone? Yes, Nishtha.
Mayank has actually raised his hand, not Nishtha.
Okay. So
Okay, Mayank, you can go
Yeah,
ahead.
so biomimicry is basically using natural things and mimicking that in man-made objects.
yeah, you are right. So can you guess that how the concept of the bird's wing is applied to the airplane wings?
Like the tips are folded over instead of being straight.
Yes, the tips are folded over. You are right. So what happens that when birds want to take off, they raise their wings, thereby creating a low pressure, and through this low pressure, the body goes up through the process of lift, and the airplane also has the same motion while taking off and landing. So this is the concept. So here is the video showing that how it goes. So here you can see that the wings. I hope it is visible. So the next question comes that is sunbath essential or not? Or we can say biologically that which vitamin we get from the sun, what are its benefit. So I'm pretty sure that you must know that which vitamin we get from the sunlight.
So Sahiti, you can go ahead.
We get vitamin D, and it helps in proper growth of. It helps for good hair. I mean, nourishment of hair, skin, and all. I think.
Yeah, you are right. Actually, vitamin D, sunlight helps in formation of vitamin D in our body. So there is a precursor present in our body, and with the help of sunlight, this precursor gets converted to vitamin D.
Okay.
Yeah.
Okay,
So
Richa, can you just go back to the previous slide, the sunbath one? Yeah, I just want to add something here. So I feel that these are two separate questions. Like, is sunbath essential or not is a different question. Which vitamin do we get from the sun? That's a different question. So yes, everyone is right here. The vitamin D we get from the sun, but it's not. Sunbath is definitely not essential for life. Actually, I would say that sunbath is actually quite harmful because vitamin D we are getting it from multiple sources even other than the sun. We are getting it from our diet, and we don't need direct exposure to sunlight for our skin to synthesize vitamin D. Like even the diffuse sunlight which is coming in through the windows, that's enough for our skin to synthesize the vitamin D. So what happens in sunbath is like you see this man, he's lying on the hammock, the sun is beating down on his skin. This is actually really bad because the sun, it also contains these things called the UV rays, UVA and UVB rays. So the beneficial effect which we are getting vitamin D, that is actually very negligible in this case. Like there is much more harm which is being caused because these UV rays, they are carcinogenic and they can lead to the production of skin cancers such as melanoma. So this is a separate question altogether. So is sunbath essential or not? No, it is definitely not essential for life. It should be avoided. You should always wear sunscreen outside. Which vitamin do we get from the sun? What are its benefits? We get vitamin D from the sun, but we do get vitamin D from our diet as well. So because of that, sunbath is not essential for life. Okay, Diksha, you can go ahead.
Yeah, thank you, Shatrupa, for adding more information. So the next is again biomimicry. So this is one another application that where we have developed the super hydrophobic surfaces. So what happens here, we can see that the water does not settle on the top of lotus leaves. So it is due to the super hydrophobicity of the leaves. So the leaf cuticle is it is a waxy cuticle. So that's why the water does not settle. And so can you guess some of the applications of the super hydrophobicity that we can use in real life? Uh, has anybody answered something in the chat?
Okay, Sahithi, you can go ahead and say. Viva has said
Uh, it,
walls.
it uses it can be used to create umbrellas, raincoats, and also in waterproof machineries. I mean, in which cannot be wet.
Yes, in waterproofing or in
Yeah,
corrosion
water
prevention. Cor-
corrosion
yeah, it can.
prevention.
So the next is that there are some more examples of biomimicry that can be used in the industry. One is the gecko leg.
Diksha, just sorry for interrupting you. Like, how do I take the attendance?
Yeah, Shatrupa, actually, I'll be the one taking the attendance because I started the meeting. So let me just make.
Okay.
Yeah, so we can continue. I have taken the attendance.